to look at DNA replication in Canvas, and the activity involves DNA replication. So we're going to do that as well. Okay. Everything in Canvas is updated. They are doing progress reports next week, so make sure that if you have something outstanding, that you bring it up to date. All right, so give me 1 minute. I am going to um, give you another worksheet for the Gizmo activity, because the one you have only has 2 pages and we have time to do the third one. So I'm going to upload an updated worksheet that has 3 pages instead of just 2. Okay. Okay. All right, so I've updated that. Like I said earlier, whatever the issue is with you all being able to log into the Gizmo has not been resolved. It's a issue on their part, not on our part. So we're going to, but I can get into it. So what I'm going to do, we're still going to do the lab, and we're going to, okay. And I have gone through, if you just turned it in, I'll check, because I just recently, before class, updated all the things that were turned in late. So if it was just turned in, I haven't seen it, but I will look again after class. All right, so what I need for you to do right now is go to, I'm trying to share right now, so give me a second for that to come up. All right, so we do this from the student point of view. Okay, so you did last class, you downloaded the one that just said building DNA, this first one. Now I want you to open this one up here. Okay, the one at the top, the one that says page 3. And what it should do is it's going to give you the same document, except this one's going to have 3 pages instead of 2. And last time I didn't think you were going to have enough time to do it all, but I did it with the last class, and they had plenty of time to do all the pages. So if you don't mind, everybody should go to if you might need to refresh your page, because I just uploaded this new document. So this one should have 3 pages. Okay, so when you open it, it should look like this. All right, and I'm going to share my Gizmo, and you're going to write down the answers, and then you're going to send me your worksheet. And because we're doing this all together and everybody's present, that is something that I expect you to have turned in at the end of class, especially since we want this to be counted towards your progress report. Okay, so everybody needs to have checked, downloaded the new worksheet so that you have 3 pages. Let me know immediately if you're not able to do that. All right, once you do that, the first 2 questions, and download it as a Google Doc so that you can write on it. And then the first 2 questions you can answer without me pulling up the Gizmo. So also have your Canvas page open as well for DNA replication. Actually, mine is closed. It's because I'm pulling up this, but you should be on the page that says DNA replication, the one we did the other day. Okay, so with that in mind, you are looking at the DNA molecule to the side. Now, they're already telling you that this shape is a double helix, so that's not the answer that they're looking for. Give a different description of what this looks like. What did we compare this to? Somebody can type it in the chat. For number one, what does it look like? What's other than saying that it's a double helix, because that's the scientific answer. What is it in layman's term? All right, Azalea said it looks like a ladder. Is it just a ladder? What else can you say about this ladder? You want to put another word in front of ladder. What kind of ladder? Malik says it's turning. That's another way we can say turning. We can say twisted. Okay, so you want to say that it's a twisted ladder. So it's not really turning, but it's twisted. Okay, it's a twisted ladder. So when we're looking at this another term that they use, it looks like a spiral staircase. All right, so for number one, you want to say either one of those, spiral staircase or a twisted ladder. Because that's important, because when we start this replication process, we got to untwist the ladder. Okay, so it's important that we know that originally it looks like a bunch of spaghetti. It's all coiled up in a ball in the nucleus. And it's just during the replication process, as we talked about when we did mitosis, it actually forms these visible chromosomes. So that's what we're going to look at. All right, the second one says, based on this picture, how do you think the DNA molecule makes a copy of itself? And that again, you can write an answer to based on what we've done already. How does it make a copy of itself? Okay. All right, so while you're looking at that, answering that question, I'm going to switch to the gizmo. So I'm going to be sharing the gizmo from here on out. So I can't do both. So y'all got to keep up. Okay. So I'll be reading the question and then expecting you all to answer on your worksheet. And there's a part where we have to take a picture. And what I'll do is when I take the picture, I'll put a copy of the picture in the chat and you can add it to your document. All right, so we've answered questions 1 and 2. All right, again, does everybody have the worksheet pulled up? Anybody not have a worksheet? Okay, good. Good. All right, so again, so I'm on the section that says Gizmo warm up. All right, it says look at the right. Um, what are the components? So on the right column, what's that name up there at the top? What are these components called? There are 8 of them. What are they called? Thank you, Malik. Nucleotides. So that's what you need to write for number 1. The components that make up DNA are nucleotides. All right. Then it goes on to tell you that a nucleotide consists of 3 parts, a phosphate, and they always represent the phosphate by the little circle. It says a little blue circle, a sugar, which is called deoxyribose, and that's by the pentagon. And then a nitrogenous base. And there are 4 different kinds. So we have 4 different colored nitrogenous base. So those 3 parts make up 1 nucleotide. Okay, and we have 8 of them right now in this column. All right, that being said, how many different nitrogenous bases do you see? So even though I have 8 of them, how many bases do you actually see? No, not 8. There are 8 nucleotides, 4. There are 4 different nucleotides. All right, I have 8 Sorry, not there are 8 nitrogenous, but let me just back up. There are 8 nucleotides, but 4 different bases. Okay, there's an A, C, a G, and a T. Okay, those are called nitrogenous bases. All right, so everybody should have put 4 for that answer. Okay, so now we're on activity A. Everybody should be on activity A. All right. It says we're going to build the DNA and we're going to follow the steps to construct the DNA. And then we're going to stop and read DNA. Okay, so this is what we have to do. We are going to drag, and now there are only 4 bases, but it's like the alphabet. There's 26 letters in the alphabet. Depending on how you arrange them will depend on what kind of words you form. We can arrange these letters in any way, shape, or form. If you all were doing it, all of you probably would have arranged them in different ways, but because we're doing it together, we're all going to have the same answer. But you can drag these over in any way that you want to. All right, this will automatically connect. Notice it keeps creating a box for you to add another one. All right, so I just randomly drag these in place. All right, so what I've done is I've created one side of the ladder. Okay, now I have to create the other side of the ladder. All right, notice the yellow boxes at the bottom. What did we learn when we did the Nearpod? What do we know about why that box is at the bottom? What do we know about the 2 sides of the ladder? Do they go in the same direction? What's the word that we need to know about why this yellow box is at the bottom and not at the top? It's not exactly inverted, but what's the word that I want to, there's another word that you have to know. All right, so what we were talking about, one side is the 5 prime to 3 prime, and then the other side is 3 prime to 5 prime. What do we call that? The 2 sides that everybody keeps telling me inverse, inverted. There's another name. There's a vocabulary word that you were supposed to write down from the last time. Anybody? Yes, you said anti something. Yes, anti something. That's exactly what it is. Anti-parallel. Y'all write that down somewhere. Anti-parallel. They are parallel to each other, but going in opposite directions. Anti-parallel. Thank you, Sarah. Anti-parallel. All right, so I'm going to do the same thing. I'm going to create the right side of the DNA molecule going from the bottom up. Now, what you need to remember is that the bases are complementary and they made it easy for you, and most activities make it easy for you so you understand they're like puzzle pieces. If I try to put the wrong piece here, it won't stay because it doesn't match. So you have to always remember that G matches C. So, and then they've rounded the pieces so that they fit. Okay. So only the piece that is supposed to match, that is complementary, will fit. So this one has a pointed edge. So this one is, so you have to remember there was a rule. Remember the rule that says that bases are complementary. AT. Okay. All right. So the instructions now. So we've created our double-stranded DNA. All right. So then it tells you to take a picture. So I'm going to take a picture. And then I'm going to put a copy of this picture in the chat, and you can add it to your worksheet. Okay, so there's a copy of the picture in the chat. You copy it and you add it to your worksheet. It says you can add it as a, you can, if you can, add it to the space on the side. Not on the side because they ask you to do something else. So they want you to write down, they want you to write these letters down. Write what's on the left side, write what's on the right side, but also add the camera picture. And you can add it at the end of the document and then label it. It tells you to label it original DNA molecule. Okay. All right, so we've done step one. We constructed our DNA molecule. All right, then they want you to take a minute to actually write down what the letters are on the left side and the right side. So with your question next to it, write down what your bases are. Yes, we're not logging into Gizmo. You are following me. I know we can't log into the Gizmo. So you are doing this based on what I'm doing on the share screen. For some reason, it lets me in, but it won't let you all in. All right, so you should have your worksheet downloaded and you're filling in these answers based on the share screen. So on number 2, it said take a picture. I clicked on the camera. I took a snapshot. And then you want to paste it somewhere on your document. You can paste it on the last page and you want to title it Original DNA Molecule. Then to the space on your right-hand side, it asks you to write down what does your left side look like, what does your right side look like, as far as you writing in the letters. Okay. All right, so I'll stay there for a minute. And then we'll look at questions 3, 4, and 5. Okay, let me see something. All right. So right here, where it says left side, right side, you need to fill in some letters. What are the letters on the left side? What are the letters on the right side? And then at the end of the document, maybe on the, or because depending on how yours will work, you might actually be able to squeeze. I saw some folks in the other class were able to squeeze that picture in and just shift the way and put it in between, right up underneath number 2. But if you can't do that, create a extra page at the end and paste your picture there. Okay. All right. So once you've done that, go ahead and answer questions 3, 4, and 5. Now, number 6 is a challenge, so that's kind of extra credit because there's a little math involved in that. So number 6 is extra credit. You want to answer this number 5, taking this picture, they've built the left side for you. You need to put the letters on the right side that should be complementary. You don't have to draw a nucleotide, but put the matching letters. All right, so I'll give you a few minutes to answer those questions. All right, so what everybody should be doing now is filling in the left and right side of this table up here, answering questions 3, 4, and 5. And again, 6 requires some math, so you can do that for extra credit. So I'll give you a few minutes to do that. We'll talk about it, then we'll move on to activity B. All right, don't forget to label your first picture because we're going to have a second picture. So label your first picture, original DNA molecule. All right. For question 3, remember that there are 2 things that make up the sides of the ladder, and the runs of the ladder are just the little sections that are in the middle. I know some of these words have similar names, so don't confuse the names. All right, what did you put for 3A? Somebody can type it in the chat. Okay, thank you, Natalie and Malik. They said a phosphate and a sugar or deoxyribose is also correct. This is more specifically because this is DNA. It is deoxyribose. That is the name of the sugar. So a phosphate and a sugar. So that's what you should have. Or phosphate and deoxyribose sugar for A. What about B? What are the runs of the ladder of the molecule made of? All right, not nucleotides, because that's what I wanted to make sure. So the whole thing is a nucleotide. So we have the phosphate in the sugar, and what's the other part called? That's why I said, let me check what y'all are writing, because they start with the same letter, but it's not a nucleotide. What's it called? The A, the T, and the G, and the C, what are those called? Yes, okay. Natalie and Zayla, so the nitrogenous bases, that's what you want to say. Nitrogenous bases, and yes, they are base pairs. But specifically, you need to know that the name of the base pairs, what they're called, are nitrogenous bases, and there are 4 of them. All right, so then for number 4 should be easy to fill out. You just write down who complements who. And then again, for number 5, you had to do a complementary strand to the strand that they gave you, which is not going to be the same as what I have up on the screen because I made this one. They're giving you practice to do another one. So you do that one. All right, so then I'm going to move on to activity B. All righty, so it says observe the enzyme. So in the little box on the left side, it says enzyme DNA helicase. What we're going to do is drag that, and I need you to watch what happens when I drag this little blue molecule over to the DNA. All right, what just happened? And you need to describe that for number one. They split. So that is the job. Yes, they disconnected. Another way to say that is they disconnected, they split. So however you wish to say that, the molecule split it into two single strands. More specifically, the DNA, it was double-stranded, and that it split it into two single strands. So you might want to say all of that. Okay. So the job of that enzyme is to unzip. Another way we can say that it unzips the DNA. So that it was double-stranded and now it's got two single strands. All right, that's number one. All right, we had in the box now, we have another little enzyme called a polymerase. So you need to watch what happens when I drag the polymerase over. All right, so it gets started and it goes in position and then it wants me to do the rest of the work. All right, so down here we're starting with a G, so I need to find a C. And then watch what it's doing. You have to answer this question. So how many polymerases do you see? Oh, reading at the wrong one. G, C, A, and T. All right, notice I have a whole nother set. So now I have to go look at the one on the other side. Oh, I skipped one. Okay. And then it finished doing the other part. All right, so now parts A, B, and C, you need to, the first one when I dragged, every time I dragged the correct nucleotide where the polymerase was, what happened? All right, the polymerase moved up the strand, and as it moved up the strand, what did it do? What did it allow me to do? The polymerase, and on one side it moved up the strand, on the other side it moved down the strand, but what did it allow me to do? What is its job? So the other one disconnected. This one does what? It's not adding new sides. What is it adding? It's adding new, uh, yeah, I guess you could call it a side. So it, yes, it added new It added new nucleotides. It did. It added new nucleotides because it added the whole thing. Okay, it added so it allowed me to connect new nucleotides to the exposed one. So we unzipped, we opened up this double-stranded DNA, and it allowed the polymerase allows you to attach new nucleotides um, to the exposed ones. So that's what its job is. So the job of the helicase was to split the DNA or unzip it or disconnect it. Y'all use several different words. This one's job, the polymerase's job, is to connect nucleotides to the exposed nucleotides. So yes, we're actually creating, so we're not just adding the runs, we're adding the whole nucleotide, the runs and the sides of the ladder. All right, B says, what happened to the DNA polymerase molecule as you added it? So did it just, how do I, so we said it moved up, but did it just glide up? What did it do? So as I added one, what did it do? I could do it, I could reset it, but it's gonna make me build the whole thing all over again. All right, let's do that. Because actually it'll be different. All right, so it doesn't matter. Oops. Um, oh, that's because it wants me to do it from the bottom. Okay. Okay, so we're going to unzip it. See, now this one, it just glided all the way up and it unzipped the molecule. With this one, what is, I need you to, it took a step, it moved. Okay, I want you to describe exactly what it's going to do. Oops. Why is it? Okay, these are the ones up here. Okay. All right, what did it just do? How would you describe what it's doing right now? It allows me to do what each time? All right, so it only moves one at a time. It allowed me to connect each nucleotide one at a time. Same thing on the other side. It only moves when each nucleotide is in place. And here again, they went anti-parallel to each other. Okay. All right. Now, letter C. How do these molecules compare to each other? Look at them closely. Look at the left strand. So we have two double-stranded DNAs. Look at them. How do they compare to each other? Thank you, Malik. They are exactly the same. They are identical. That's the answer you should have for C. They are exactly the same. So it opened up. So remember when we did mitosis, we said DNA replicates, so it makes an exact copy of itself. So at the end of mitosis, the two cells were had exactly the same information. So we just went through replication. We replicated the DNA. All right, the next one says, click on the camera, take a picture, and label this daughter DNA molecule. So I'm going to do the same that I did before. I'm going to copy this. I'm going to put it in the chat, and you are going to add this to your worksheet. So label this one. This is number 3. All right, so now they want you to look at this one. And actually, because I rebuilt it, we were supposed to use the same ones as we did before, and I did not write down it. But what you need to say is it would have been identical had I not rebuilt it. So what I should have done was rebuilt this one the same way as the first one, and I didn't. I randomly put this one together. Um, but what should have happened was the one that you built the first time, had I not reset it, they should be exactly alike. And actually, let's do that. Let me go back and so don't copy that picture. Let me go back and create a picture that's going to be accurate. So let me look at your first one. Okay, let me blow this up. Okay, don't copy that one. You can um, answer the discussion question, but I'm going to recreate the one that you should have so that you can answer the question correctly and say that they are exactly the same because they should be. And that was the whole point of replication. So let me put this over here and let me replicate this the way it should be replicated. All right, so in the meantime, um, answer the other question that you should have, and I'm going to create the right picture for you. So, okay. All right, so ignore that second picture that I sent you. Just make it go away. And I'm going to create the picture that you should have so that you can accurately see what should have happened. can I delete that one? All right, so in the chat, I deleted the other one. So download this one. So when you look at this one, it should look exactly like the one you had the first time, except now there's two of them. You have replicated the strands. Okay, so the answer to the question for number 3, it says, how do these molecules compare with the original? So you were to label this one, when you did the camera shot, you were supposed to say daughter DNA molecules. That's how this one should be labeled, daughter DNA molecules. Question 3. And then you should say, how do these molecules compare with the original one? So in the chat, if we scroll back and look at the first one, thought I made it disappear. This one should look exactly like the single-stranded one. I mean, the double-stranded one we did at the beginning. I think I deleted the wrong one. Yeah, I did. So hopefully you got the first one out of the chat. Because I deleted the wrong one. So let's delete this one. Delete. So there's only one. Okay. All right, everybody good? All right, so for this one, you want to go ahead and answer questions 4 and 5. And then we've completed this activity. So what you want to do when you're finished with this is go ahead and upload it. And again, we did this together. So everybody should finish today and upload it to your, oh, wait a second. Okay, I do have to fix this. All right, so while you're working on that, I need to fix it so that you can submit it. Because right now it says no submission. Okay. All right, so when you're finished, you might have to refresh your page, but then you should be able to submit. All right, when you finish doing that, I need you to go back to the, so on when Monday's a holiday, so I see you all on Wednesday, because Wednesday is going to be a Tuesday schedule. So your Monday schedule will be Tuesday and your Tuesday schedule will be Wednesday. So I see you on Wednesday. And on Wednesday, we're going to have a quiz. Okay, so on Wednesday, we're going to go over the information in the DNA module. So we need to know the first 2 units. We haven't done protein synthesis yet, but that's our next step. We did all of this so that we can take it to the next step. All right, so not some, you need to know who Watson and Crick are. You need to know that the difference between RNA and DNA. And you need to know that there are 3 types of DNA. You need to go through this little activity here. So study that because this will be on there. Do the review. All right, so what we did today, activity, is we demonstrated this process. Okay, we went from a double-stranded DNA, we replicated it, we ended up with two double-stranded DNAs. So go through this process. This also talks about the enzymes helicase and polymerase, and they give you an extra one there. All right, and then come down here and make sure that you have done this, because this will also be on the quiz. So I'll give you a few minutes to finish what you're doing, and then practice that, and then I'll put up correct answers, although you can check your answers. So again, quiz on Tuesday, going through these first two units. As always, read this part down here as well. So our next step going forward, now that we know how to replicate DNA and while we know what RNA is, our next step is to do protein synthesis, but we'll do that next week. Okay. All right. So what everybody's doing right now is making sure if your body was present today, that means you should upload a worksheet today. After you finish that, come to your Canvas page and do that activity right there. All right, I'm going to unshare, so I haven't done attendance yet, so I need to do that. All right, so again, everybody's worksheet should be uploaded, and when you finish that, you need to be on Canvas uh, doing the matching. Okay, the only person. All right. Also, if you were doing the extra credit color activity, please make sure that that's in today as well. Landon, you went through a lot of trouble. I appreciate you. Okay, so I repeat, everybody should have their worksheet uploaded. Everybody's worksheet should be finished, saved, and uploaded. So far, I only have two, three. Okay. All right. So again, while you're reviewing, make sure that you know uh, the base pair rule. Make sure you know what that is. Which ones bond to which ones. All right. Very good, Natalie. She got the challenge question. See that? Okay. And again, make sure that you put the correct pictures on the worksheet. So the second one so when you look at them, and you need to look at them to make sure that your original and your daughter DNAs look identical. Mm. All right. Um, Make sure that you understand too, because I'm looking at answers for number two on the worksheet, that when it asks you based on the picture, how does it make a copy of itself? You want to, um, you might even go say as far as say during the process of mitosis, the strands unzip or separate and make an exact copy of the unzipped portion of the DNA. Because actually what we're going to look at later on is that the whole thing doesn't unzip, just a certain portion of it unzips. And that is shown to you in Canvas in the little replication box. So look at that box again. Let me share so that I can point that out. Trying to look at these. Okay. Alrighty. Sean, I don't know if you meant to leave questions for, okay, never mind. Okay, I got it. I got it. Never mind. Okay. Sometimes when y'all type the answers, you type it so close to the question that it looks like a continuation of the question. All right. You all keep making sure that you upload your worksheets. I'm going to stop grading for a minute so that I can point something out to you that you should be looking at. All right, any questions? Any questions? Now is the time to ask the question. So again, the activity was to demonstrate replication. We replicated a DNA molecule. Okay. All right, make sure that you go back and you read these boxes, but the box in particular that I was looking for is on the other page. No, it wasn't. I wanted this one. Okay, yeah, yeah, this one. All right, so make sure you read through what is happening here as far as All right, so the DNA strand split in two. Each half of the parent serves as a template. Template means that that's a surface where something else can be added to it. Okay, it serves as a template for the new strand. And the results is the new daughter cells having both the old and the new DNA strands. So the two sides that open up are the old ones. The strand that's inside is the new one. So each new one has an old strand and a new strand. Okay, and so that was what the activity that we just did was showing. Okay, what this was.
Within the nucleus of every cell are long double-stranded molecules, DNA. DNA is a structure which exists in the form of a ladder twisted into a spiral or double helix. The millions of rungs in the ladder are composed of nucleotide base pairs that interact, either thymine T with adenine A or guanine G with cytosine C. DNA duplicates itself in the process called replication. As the double helix unwinds, each strand becomes the template for a new strand. Unattached nucleotides move in and link up to their complementary nucleotides, A to T and C to G. Two new molecules of DNA result from this process. Each is identical to the parental DNA.
Oh, whoops. Sometimes it's hard to get these things to stay put. All right, if you haven't done this check, I always do those two backwards. It unwinds. It unzips. Okay, there we go. All right, so make sure you have those, that you know the order of the process. All right, so this matching activity and the other matching activity on the other page, they'll be on the quiz, as well as knowing this information in that box. Okay, any questions? Any questions? All right, so if you've uploaded all your work and you've got the information you need from here, from Canvas, you're good to go. You have a great weekend.